I'm Daniel Levine, and this is the Bio Report.
Alzheimer's disease remains one of medicine's most intractable challenges. While existing therapies may address individual features of the disease, such as amyloid or tau, they have yet to reverse the progressive cognitive decline, inflammation, and neurodegeneration that characterizes it. Evinco Therapeutics is developing nanoscale natural killer cell-derived particles that carry proteins and microRNAs to target the neuroinflammation underlying Alzheimer's. Alan Trounson, founder and CEO of Evinco, discusses the company's multifaceted therapeutic approach, how these particles are designed to cross the blood-brain barrier, and their potential to modulate neuroinflammation, support the brain's immune cells, and promote amyloid clearance. Alan, thanks for joining us.
Good to be with you, Danny.
We're going to talk about Alzheimer's disease, Evinco Therapeutics, and your efforts to develop natural killer cell-derived extracellular vesicles to treat the condition. Let's start with Alzheimer's disease, though. This has been a difficult area for drug development. What makes it particularly challenging compared to other major diseases?
I think all of the neurological diseases have been difficult to develop or challenging for therapeutic development. And the exact cause of Alzheimer's is still not really known. It begins very slowly and involves a loss of cognitive function. And then you see amyloid plaque formation, tau tangles in the neurons, inflammation and memory loss. Targeting sort of single components of this condition really hasn't led to very high rates of recovery, slowing the disease, but not recovery. And the animal models really only approximate rather than adequately mirror the human condition, and lab models really haven't been that informative either for translation. So it's really that by chance or serendipity will probably open new therapeutic approaches, and it seemingly has.
Do you think we've been targeting the wrong biology, or is it limitations in trial designs, or is it intervening too late in the progress of the disease?
I think the condition is too complicated to just target single components. Targeting the amyloid or targeting the tau seems to have some benefit in slowing the condition, but doesn't seem to provide any recovery. And we haven't really been trying to treat inflammation, which is an underlying component, and I think maybe an initiating of the whole process, either a viral or physical initiation of damage in the brain that then cascades into the Alzheimer's condition. And if you look at traumatic brain injury, for example, you see the same kind of changes in a more accelerated way that you see in Alzheimer's. So I think, some kind of injuries happens, and then you get an inflammation, and then you get these other conditions happening, the plaque formation and then the tangle. So this is pretty complicated, and we really haven't tried to adequately address the immunology that's the underlying issue of inflammation. And if you don't do that, I think the condition continues and doesn't reverse.
How do you think about the limitations of traditional small molecule or antibody therapies in the context of neurodegeneration?
Yeah, I think they're useful because, they are addressing some of the component parts of Alzheimer's, but they're not addressing the entirety of it. Um, Small molecules will target signaling pathways, but leave other pathologies intact. Might get off-target toxicities as well with small molecules. When antibodies are, are, for example, targeting extracellular amyloid, and I don't think that's the entire condition. So you're only treating component parts of it and not the entirety. And we need to rethink the strategies to enable multiple actions or combination therapies that might be needed for a more comprehensive response.
Given that, to what extent do you think the drug developers have really broadened their mechanistic lens when looking at this disease?
Well, I don't think they have adequately done that. I think the immunology has been left out, and I do think that microglia are playing a pretty important role here because they're a very key component of the immune system in the brain, and they haven't really been treated, I think, in a way that's completely appropriate. So if you have an immune component in the brain which is not doing what it's intended to do to clean up the abnormal and dying cells there, and also removing them plaque and other component parts, and it's not doing that, then I think you have to address that, and I don't think it's been adequately addressed. And that's why I think there's probably some merit in looking in more deeply at the biology and using some biological approaches.
There has been growing momentum behind cell-derived and biologically complex therapeutic. What's driving that shift now?
I think the interest now that there is an inflammation component there, and to most neurological conditions, I think that's clearly the case. So, it's not, you can use drugs to try and dampen inflammation, but the immune system's evolved to treat that, to control inflammation, and it's better served to do that in a complex organ like the brain. So I think there is some interest. There's, of course, interest from the stem cell communities in introducing stem cells to the brain, but I don't think that's going to be as useful maybe as Parkinson's disease, but it could be a component part, and there's even interest in using extracellular vesicles from neurological stem cells that might be useful there. But again, I'm not convinced that those approaches are matured enough yet to be to be helpful.
So, what's the potential here for a type of paradigm shift that we saw happen in oncology with cell therapies?
Yes, I do think there's a need for paradigm shift, and I think the field has shifted because the discussions I've had with the senior researchers in Alzheimer's and other neurological conditions is the recognition that inflammation is a pretty primal component part that needs to be addressed, and that an immune approach may well be much more useful than simply small molecules, or antibodies. I do think those antibodies and small molecules could be useful, but in combination therapies, perhaps with more biological approaches.
You're not alone in pursuing Alzheimer's disease as an inflammatory disease. What's the case for that perspective right now?
Oh, the case is that you see all the inflammatory markers if you look at these conditions, you find all the inflammatory markers that you'd expect to be increased there, and you can find them very early in the condition, and increasingly becoming even more amplified as the condition worsens. So I don't think there's any doubt that inflammation is playing a process. And if you look at brain sections from people who have died, and clearly the brain has got an inflammatory response there. So that there isn't any doubt that inflammation is present, and it's whether you can control that, because in inflammation you get function of the immune system, which is not really what's required for a healthy tissue. You have to actually get the immune cells back in a state to do the proper job, and they are not doing that in an inflammation environment.
Evinco is developing natural killer cell-derived extracellular vesicles, or NKEVs, to treat Alzheimer's disease. What are NKEVs?
Um, NKEVs are extracellular vesicles, which are very tiny. They're 50 to 150 nanometer particles that are surrounded by a lipid bilayer, and they contain a cargo which is representative of the cell that they come from. So, a natural killer cell EV will contain microRNAs and proteins that are present within the natural killer cells, and they're usually released from the cells to communicate messages to other cells. We're interested because they're produced in abundance from natural killer cells, and we've been very interested to see the really the work of the company NKGen, giving patients pretty massive doses of NK cells intravenously and getting a really surprisingly good response in patients with Alzheimer's disease. So we thought those natural killer cells wouldn't be crossing the blood-brain barrier, but extracellular vesicles, because they're so small, can easily pass the blood-brain barrier. So we became interested in those to see whether these NK vesicles would actually do the same job as natural killer cells, and at least in vitro, these EVs are quite remarkable in the way they change microglia and astrocytes in their abilities to remove amyloid plaques and metabolize it away, but also suppress inflammation.
There's been interest in NKEVs as potential anti-cancer therapies. What makes them well suited as treatments for Alzheimer's disease?
I think this was a serendipity observation by the company NKGen, who were treating patients who'd had radio and chemotherapy, treating them because they're concerned that the patients would be exposed to lethal infections. And by chance, they treated an Alzheimer's patient, and then went on and got quite a remarkable response in that patient, and treated some others and found also they responded, and then just started work in Mexico, then back in the US. It's shown a kind of 90+% response in these patients. So it's a serendipity observation, I think, because I wouldn't have expected that. I don't think the company did. I don't think other scientists thought about natural killer cells in this regard. But serendipity plays a very big part in science, as you know, in making these observations from something that you were looking for a different response and you got one that you didn't expect, is often the basis for a big change. And that's what I was interested in, because we were making natural killer cells for cancer, as you said, for treating cancer. And I thought, well, maybe they're producing something that gets across the brain in quantity that would actually impact on the microglia and potentially the astrocytes and neurons to have a beneficial effect. So that's the underlying hypothesis, and that's what we've been following.
How well understood is how they might act in a patient with Alzheimer's disease? Are they expected to create immune modulation or protein clearance or neuroprotection?
They do all of these things. So what they do, we can show that these extracellular vesicles will stimulate the microglia to clean up the aggregated amyloid plaque type material, gobble it up, like a cleaning process going on, and then metabolize it away. Other cells can't, EVs from other cells can't do that. For example, mesenchymal stem cell EVs don't do it. And even neurological stem cell EVs, they take it up, but they can't metabolize it away. So, so they have that property. They suppress inflammation in microglia and in astrocytes, so they change those back to a more normal type of cell under that's operative in the normal conditions. And I think they have an impact on the tau phosphorylation as well. We're looking at that in some combination treatments with colleagues in collaboration.
You mentioned because of their size, they can cross the blood-brain barrier, unlike the cells that produce them. How would they be delivered?
Well, they could be delivered intravenously, but I think extracellular vesicles are quite commonly delivered nasally in a nasal spray. So EVs from mesenchymal stem cells, which are in clinical trials, are delivered that way, and you can see that data in many of animal models. So that's the route that we would prefer to go, and that's the route that we're exploring. The delivery via the nasal route would allow this material to be delivered very simply without any hospitalization. And because EVs can be freeze-dried and shipped around at room temperature, they could be, put in vials and then reconstituted at home and then introduced into a nasal delivery adapter and used very simply. So this makes it very, very, user-friendly, if you like, and it gets rid of a lot of the problems of cell therapies, having to move them around in frozen states and thaw them and freeze them and thaw them and so on. So it is, there's some huge advantages to that.
And in terms of manufacturability and scaling manufacturing, how does the production of these compare to, say, the production of cell therapies?
Yeah, it's pretty straightforward because if you're using, say, mature natural killer cells, they can be made in very large quantities. There are companies that make them in 200 liters or even 500 liters, and that would make hundreds of thousands of doses of these EVs for patients at very low cost. So cost would be very small compared to cell therapy. Cost of goods would be extremely small compared to cellular therapies, and the volumes would be very large. So, so even from one manufacturing run, you get a very large number if you went into such large-scale manufacturing. Presently, we make them in a relatively small amounts, but that's because we're doing the research. But the companies that are set up for a natural killer expansion get into very large quantities. So we don't see the manufacturing as being a very difficult problem at all.
And what's known about the NKEVs from studies you've conducted to date?
Well, as I said, we can see the properties of inducing the brain's immune cells, the microglia, to clean up the amyloid very effectively, very efficiently. It stimulates a massive increase in their ability to do that, but they also in their ability to metabolize it away. So it's they can continue to do that. Whereas in some other EVs from other cells, if the microglia take up large quantities of amyloid, it's toxic and they die, and at least in vitro in the lab. We see the effects of reduction in IL-6 and other markers of inflammation in astrocytes and microglia, and we actually can get the microglia to alter their properties to be much more like the normal microglia rather than the inflammatory types. And so I think they also will have an impact on tau phosphorylation, but we're still looking to get that data from our colleagues. But the work from NKGen showed a marked reduction in phosphorylated tau in their studies when they gave these very big doses of NK cells to patients. So we expect that to have them to have an impact on the tau phosphorylation and the tangles as well. So all of those are really strong benefits. Now, whether they can have a neurotrophic effect to stimulate neurogenesis, again, I don't know, but there are other components of other cells that can do that, EBs from other cells. So you may want to follow the NK cells with a different type of EV, or perhaps another combination therapy of another type to stimulate neurogenesis and get you back much more towards normal. But the clinical work of NKGen shows that a lot of these patients are really quite severe Alzheimer's have progressed back towards normal quite dramatically.
And what's the clinical path forward? Do you expect to advance this into the clinic independently, or are you seeking a partner?
Oh, no, we would think this was something that we would partner with, and we have a large pharma helping us design the studies at the present time. So that's pretty useful to have their advice as we're conducting these preclinical studies. And I expect that they or other pharma or drug companies would be very interested in a product like this. It really has applications in other neurological conditions as well, so it's likely to be very interesting to them. Yeah.
Well, how are you thinking about other conditions beyond Alzheimer's? Are you looking at neurodegenerative or even systemic diseases?
Now we're looking at, for example, traumatic brain injury, because that's such a massive problem with no treatment. It's costing the globe around 1.1 trillion at least a year. A recent publication in Lancet reported that, and there's no treatments for a traumatic brain injury. And we think it shows exactly the same sort of marker changes as Alzheimer's in a more accelerated way. So we think there's good reason to explore traumatic brain injury as a very major market to help in that condition. And of course, Parkinson's disease is something that's being again treated with NK cells by the company NKGen, and they've had some responses there as well. So there's strong interest from the Parkinson's patients for us to also include that at some stage. So that, for a small company, Danny, that's quite a lot of work to do. So right at the moment, we're concentrating on Alzheimer's disease, but we see real relevance in traumatic brain injury, and see that this may well be applicable to other neurological conditions as well.
As you think about possible clinical trials for Alzheimer's, are cognitive measures sufficient, or do we need better biomarkers and earlier intervention frameworks?
Well, we need biomarkers for sure to assess the changes, and biomarkers have become, well established now. There's a lot of biomarkers for inflammation and for amyloid, tau, and other component parts. And if you look at traumatic brain injury, they also have a lot of biomarkers that can be followed. And if you look at those biomarkers that they use they're also really critical in Alzheimer's as well. So we'd want to use all those biomarkers to see those changes and see which component, if we really, are we addressing the whole of the condition or are we only addressing a component part of it? So those biomarkers are critical. Of course, cognitive changes are what the patients need, and they'll be very interested in that. And clearly that's been happening in those clinical trials for NK cells. So we hope and we'd expect that maybe we'd see that those cognitive improvements will happen as well in these studies, and they need to be they need to be they need to change because it's really what it's really what the patients need.
Early in this discussion, you seem to be making a case for combination therapies as opposed to monotherapies in treating Alzheimer's. Do you see this as being a foundational component for a combination approach?
Yes, I do. I think that's what the pharma colleagues are thinking as well. If you can actually address some of the issues of the condition with EVs, then you may have a much better effect with your antibodies or your small molecules. For example, the bleeding in the brain is something which is a concern for the antibody trial work. And if you could actually, remove the amyloid from around the vasculature, you probably have an improvement in the brain bleeding that happens with the antibody, the administration of antibodies. So yes, I see the combination therapies as something that will evolve in this area, and I think they will be necessary because these are complex conditions, and we need to be able to treat the whole of it rather than just a component part. And I think the EVs, and as a biological entity, will be helpful in getting more broadly across what's needed, but I don't think they will provide the complete answer. And I think other combinations of either antibodies, small molecules, or EVs from other sources will actually end up being what we use in the in the fullness of time.
And from a regulatory standpoint, do you see NKEVs posing any additional challenges?
No, I don't see that they have many challenges there because, as far as I can tell, there's been a lot of clinical trials on EVs, in animals and in the humans now. There's several hundred clinical trials going on, and I haven't really read of any um, negative effect of EVs at all in patients or in animals. So I doubt there'll be any concern for risk with um, the delivery of EVs, and if it's a nasal delivery, you don't have any associated medical administration risks. So I don't think the risk will be very small, if any. And if there's a benefit, then I think there will be a rapid movement for accepting these as a component part of the therapies for Alzheimer's and other conditions. I don't think there'll be any real problem with the regulators in this area.
Some listeners may remember you from your time as head of the California Institute for Regenerative Medicine. In that role, you had visibility into early innovation across the regenerative medicine landscape. What did you learn about the translational challenges these therapies face in reaching the market?
I think the problems for cell therapies were much larger than I think were anticipated in the first place. Biological cell therapies are complicated and have taken a long time to get through the preclinical and clinical processes, but they're arriving now as treatments. So stem cell derivatives are being used for treatments of a large number of conditions now. So they get there, but there tends to be a very difficult process, and in many respects, the immune system has played a difficult role for cell therapies because the foreign cells from a donor or foreign cells are reacted to by patients, and so the immune system has really been a barrier, if you like, to getting those therapies in place. The benefits of extracellular vesicles is they're not recognised by the immune system at all. They can't see any response, the T cells and no antibody responses that we've seen. So I don't think they express histocompatibility antigens on the surface of the extracellular vesicles. So take out that problem of the immune system, which is a pretty major one. And I think these things will then travel much more quickly as a non-cellular therapy. But I think the progress in stem cell therapies broadly has been really fantastic, and I think they're providing some very much needed therapies for a large range of conditions. But it's been taken a long time and much longer than I think that I expected or we all expected in the early 2000s when we first started in on that. But I'm encouraged by what's been happening in the field.
Now, what do you think it'll finally take to bend the curve on Alzheimer's and other neurodegenerative diseases?
Oh, I think if we can show some changes here when we influence the immune system in the brain, particularly the microglia, then I think the field will change, and I think it's actually changed already now. When I talk to senior scientists in neurological research and Alzheimer's, they all recognise inflammation is something that needs to be addressed and hasn't really been adequately addressed up until now, and they're very interested in what we're doing, which it might have been surprising, I guess, because it's quite radical because it's a very different approach, but it's based on a serendipity observation, and often they produce changes in the field which are really helpful, particularly in an area where it's been difficult to make progress. We've made progress in understanding the disease of Alzheimer's and others better, but we haven't been able to construct something that's of real major benefit for the patients, and that's the challenge. And if we can show that there's some impact of this type of approach on the brain's immune system, it will change the field, I think, dramatically, as we saw in cancer. And I hope will make a very large difference to the patients and the benefits that will be emerging from that change in the field.
Alan Trounson, founder and CEO of Evinco Therapeutics. Alan, thanks so much for your time today.
Thanks, Danny. Been a pleasure.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.